we having a little bit space, you will write RFLP. There is a technique RFLP, restriction fragment length polymorphism. Restriction fragment length polymorphism. What is RFLP? I told you that restriction endonuclease cut palindrome region, and under Human Genome Project we defined the complete genome. At this time, if I ask you, do you know the nucleotide sequence of beta globin gene, you will say yes sir, we know. We will check and we will tell you that this is the normal sequence of beta globin gene. You know that what is the mutation in sickle cell anemia, you already know that this is the mutation in sickle cell anemia. So at this time you know that most of the nucleotide sequence of genes, you know nucleotide sequence of nucleotide sequence of most of the genes, you know the mutation in the genes, and you already know that this mutation lead to this illness. So whatever we are doing in the lab, that we are doing very superficial work. All the work has been done. Human Genome Project started in 1990, saying that we multiple countries came together and they said that okay, we will meet together and we will try to uh, determine the nucleotide sequence of human being. And they took target of 20 years and they started with the chain termination method. Later on they shifted to new generation sequencing, and it was completed in 2003. 90% of genomic sequence has been was determined by till that. So this is Human Genome Project. Under Human Genome Project we know the nucleotide sequence of each and every normal gene. I take simple example of beta globin gene. Suppose we found, I am not telling you the exact sequence of beta globin gene, nucleotide sequence of beta globin gene, I am giving you the concept. No need to remember, just remember the concept. Suppose this is normal beta globin gene. In normal beta globin gene I found two palindrome, GGCC, 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 GGCC. Suppose a normal beta globin gene is having two palindrome, and we all know whenever restriction endonuclease act on palindrome, they cut this palindrome. They cut this palindrome. So we already know that normal beta globin gene, if you take normal beta globin gene, it is having two palindrome. If you will put restriction endonuclease, it will cut at two sides and three fragments will be formed of this size, of this size, and of this size. We already know. We also know that in sickle cell anemia, there is point mutation in beta globin gene. We already know that in sickle cell anemia there is point mutation in beta globin gene, and we also know that due to this point mutation there is point mutation GG CC, GGCC, GACC, GGCC. Yes, this point mutation occur and this palindrome is lost. This palindrome is lost. We found that in sickle cell anemia there is point mutation, and due to this point mutation this palindrome is lost. Only one palindrome is left, and when restriction endonuclease will act on it, restriction endonuclease will cut at only one site and only two fragments will be formed of this size and a different fragment of different size. Because after mutation this is not a palindrome, restriction endonuclease will not be able to cut it. So these fragments which are produced, these are called restriction fragments. Means on any gene, if restriction endonuclease act and it produce fragments. Those are called restriction fragment. In normal beta globin gene, three fragments are produced. In sickle cell anemia, only two fragments are produced, and their length is changing. That's why the name of technique is restriction fragment length polymorphism. So RFLP, in this technique, we can detect point mutation. We can detect point mutation means even a single mutation, single nucleotide change is there that can be detected. How it can be detected? I know that in normal beta globin gene, restriction endonuclease will cut at two sides, three fragments. If you will do electrophoresis, these fragments will move according to their size. In this, three fragments will be, three bands will be formed. In sickle cell anemia, yes, two fragments are there on electrophoresis. One fragment will be on the same site, and the other fragment at different site. So you can easily say that in electrophoresis, movement depends on size, shape, and charge. Now two these fragments are moving according to their size, and we found that in normal beta globin gene, due to restriction endonuclease, three fragments are obtained, and these three fragments produce three band on electrophoresis. But in sickle cell anemia, two fragments are obtained, and on, if you will do electrophoresis, you will find different electrophoretic picture. Now, till now we know all this information. I have not done any experiment. Now I start experiment. A clinician suspected that this patient may have sickle cell anemia. Patient was sent to me. I want to diagnose that patient is having sickle cell anemia or not. I took the sample. I removed beta globin gene. I acted restriction endonuclease. Restriction endonuclease may cut at two sites or one side. I don't know. Again, I repeat, I don't know. I took the sample, I took beta globin gene, I put restriction endonuclease. If person is normal, this restriction endonuclease will cut at two sites, and on electrophoresis, three bands will be formed. If person is having sickle cell anemia, one palindrome is lost, restriction endonuclease will cut at one side, two fragments will be obtained, and electrophoresis two band will be obtained. So it's very simple. Whenever you want to diagnose sickle cell anemia, we take the sample, beta globin gene, restriction endonuclease, electrophoresis, three band normal, two band sickle cell anemia. If I ask you a question, please do reply. I gave you this electrophotogram. What you will say? The patient beta globin gene is normal or having thalassemia? Sorry, having sickle cell anemia. Please do reply. Please do reply. If I give you this, normal. Yes, but you will say, sir, we can't visualize this DNA. DNA is colorless. Yes, after that you did electrophoresis. Now I can't see that how many bands are there because DNA is colorless. If DNA, yes, you can't tell me. You can't tell me because you can't visualize DNA. What you will do to visualize DNA? To visualize DNA, I do southern blotting. Means after RFLP, I will do southern blotting. What is southern blotting? I will take this electrophotogram and will take a nitrocellulose paper. I will keep these two side by side. Impression will come on nitrocellulose paper. Now I take, yes, all these impression. I don't know the patient is having 3 band or 2 band. I took sample, I took beta globin gene, restriction endonuclease, electrophoresis. I don't know how many bands are there because I can't see DNA. DNA is colorless. That's why to visualize DNA, I do southern blotting. In southern blotting, I blot the DNA. Yes, these electrophotogram. I take electrophotogram and I take nitrocellulose paper, put side by side. Impression come on nitrocellulose paper. Now I take radioactive complementary probe. What I take? First thing I did blotting. Yes, shift to nitrocellulose paper. Then we will take radio labeled probe. We will take radio labeled probe, complementary probe, radioactive probe. I will flow over this nitrocellulose paper. I don't know how many bands are there. Till now I am not aware. I again tell you what I did till now. I took sample, beta globin gene, restriction nuclease, electrophoresis, took nitrocellulose paper, put over it, flow complementary probe, radioactive complementary probe. After that, I will wash the remaining complementary probe and I put X-ray plate. I will put X-ray plate. Auto radiography. Yes, next day, yes, these complementary probe will emit radioactive waves. If complementary probe bind at 3 sites, they would release the radioactive rays at 3 sites, two white bands on X-ray plate. If 2 bands are there, yes, 2 white bands on X-ray plate, then you can detect the patient is having sickle cell anemia or not. Now to visualize DNA, we do southern blotting. So RFLP is not complete itself. Means after RFLP, we do southern blotting to visualize DNA. Yes. Now valid question. Yes, that it can detect point mutation only those mutation which affect the palindrome region. This is the limitation. Question is asked, what are the limitation of RFLP? Yes, limitation of RFLP. It can detect only those mutation which affect palindrome. Yes, RFLP can detect only those mutation which affect the palindrome. So second is radio labeled probe. Third is X-ray plate autoradiography. So simple. Now you can say yes. RFLP can detect only those mutation which affect the palindrome, otherwise you can't. So simply RFLP, to complement RFLP, we also have southern blotting. So now I can discuss very simply that patient came to me, I want to detect point mutation. We know that this point mutation affects the palindrome region, sickle cell anemia. We know already know that normal beta globin gene, electrophoresis, 3 bands here. In sickle cell anemia, 2 bands are there. Now I took sample, I took beta globin gene, restriction nuclease, cut. I don't know how many fragments produced, but I will do electrophoresis. On electrophoresis, if 3 bands are there, normal. If 2 bands are there, sickle cell anemia. How we will visualize this DNA? We will visualize DNA by southern blotting. What is southern blotting? First of all, we will shift the impression on the nitrocellulose paper. Now impression came on nitrocellulose paper. Now what happened? On nitrocellulose paper, we will take complementary probe, radioactive complementary probe, flow over this nitrocellulose paper, and we will wash the remaining complementary probe, radioactive, and we will put X-ray plate. When we will put X-ray plate, these radioactive probe will emit the rays, and if 3 bands are there, they will emit rays at 3 sites. If 2 bands are there, they will emit rays at 2 sites, and there would be 3 bands or 2 bands. If you will check the this X-ray plate, if 3 bands are there, then it is normal. Two bands are there, it is sickle cell anemia. This is RFLP complemented by southern blotting. To visualize DNA, we do southern blotting. To visualize, yes, RNA, northern blotting. Yes. Visualize protein, western blotting. Southern blotting, northern blotting, western blotting. Mnemonic is snowdrop. Southern blotting for DNA, northern blotting for RNA, western blotting for protein. So you can detect one mutation at a time by southern blotting. But you will find that sometimes patients are having multiple mutation, then you can detect by microarray. Question: microarray can detect multiple mutations. Multiple mutations can detect multiple mutations at a time. Microarray, in microarray there is DNA chip. There is DNA chip, just like glass slide. On this DNA chip, there are thousands of microscopic spots. Thousands of microscopic spots. Thousands of microscopic spots. Each microscopic spots have so many complementary probes. Means I took a glass slide. Actually, this is not made by me. This is made by very high robotic technique. Suppose there is a DNA chip. There is a very small chip. On this DNA chip, there are small microscopic spots. What does microscopic spots means? You can't see them by naked eyes. Yes, you can see, visualize them by microscope. So on each spot we have attached complementary probe. Suppose at position 1 we have attached complementary probe for normal beta globin gene. At position 2 we have attached complementary probe for beta thalassemia. Yes, at position 3 I attach complementary probe for normal alpha globin gene. We all have knowledge that this is the nucleotide sequence of alpha globin gene, this is the normal sequence of beta globin gene. We have complementary probe for normal alpha globin gene, we have complementary probe for normal beta globin gene, we have complementary probe for beta thalassemia. We already defined that beta thalassemia, this mutation occur, and due to this mutation nucleotide changes from this to this, and we made complementary probe to the changed nucleotide sequence, and we attached at position 2. At position 4 we attached complementary probe for alpha thalassemia. In this way, I attach complementary probe for normal gene, mutated gene, normal gene, mutated gene. Suppose at position 10th, I attach complementary probe for cystic fibrosis. I have little bit modified to make simple, yes, but concept is correct, yes. So I attach complementary probe for each and every normal gene or mutated gene. Now I give you cell and I ask you, can you tell me how many mutations are there in this cell, or which genes are normal, which genes are mutated? You will say, yes sir, I can easily tell you. Yes, you will take the sample, you will take the cell, you will extract the nucleus, you will cut the DNA at the level of gene. Gene may be normal or mutated, I don't know. I took the sample, I took the gene, I attached fluorescent dye. I attach fluorescent dye to each and every gene. It may be normal or it may be mutated, I don't care. Yes, I attach fluorescent dye to each and every gene. Now I put this sample on this DNA chip. I put this sample on DNA chip. Now this sample will be flowed over DNA chip. Now Yes, if patient is having normal beta globin gene, then this complementary probe of normal beta globin gene will bind to the normal gene of the person. If patient is having alpha thalassemia, then complementary probe for alpha thalassemia will bind to the mutated gene of person, means alpha thalassemia gene. And this patient, this second position and third position would be vacant. Suppose this patient has also cystic fibrosis, then mutated cystic fibrosis gene will bind at position 10th. So if genes are normal, they will bind to the site of normal gene complementary probe. If mutated, they will bind to at multiple site, and they will bind to that site of mutated complementary probe, means complementary probe for mutated gene. In this way, each and every gene, it may be mutated, it may be normal, will bind to some site, and then we will wash the sample and we will put the DNA chip in the computer will detect, okay, fluorescence is coming from position 1. Position 1 means beta globin gene is normal. Fluorescence is coming from position 4, means patient is having alpha thalassemia. Fluorescence is also coming from position 10th, cystic fibrosis. So you can detect multiple mutation at the time. It will tell you this gene is normal mutated, means a list would come that this gene is normal, this is mutated, this is normal, this is mutated, means multiple mutation you can detect by microarray. Please do reply understood this microarray. So in any question, wherever you find multiple mutation can be detected by microarray. Clear? Microarray is not only for DNA. It can determine global pattern of gene expression. Can determine global pattern of gene expression. What does it mean? I told you that in our genome there are 25,000 genes. There are 25,000 genes. Yes. If I ask you, I give you a cell and I ask you, can you tell me out of 25,000 genes, how many genes are expressed in this cell? You can tell me that how many genes. Yes, I ask you how many genes are expressed in this cell. You told that yes, sir, in this cell, these out of 25,000 genes, 2,000 genes are expressed. I can determine by microarray. Means I can determine the number of expression of gene by microarray, and this is very good technique to determine global pattern of gene expression. What does it mean? Global pattern of gene expression means out of 25,000 genes, how many genes are expressed. If I ask you, can you tell me that from each gene, how many messenger RNA are produced? You will say yes, sir. Out of 25,000 genes, 2,000 genes are expressed. Gene number 1 produce 3 messenger RNA, gene number 2 produce 5 messenger RNA. Means you can tell me so much minor detail. This is called global pattern of gene expression. So we can determine global pattern of gene expression by microarray. It helps us to determine the expression of gene into different cells. Suppose this is normal cell, this is cancer cell. In normal cell, gene number 1, first gene produce 2 messenger RNA. In cancer cell, same gene produce 4 messenger RNA. Okay, in normal cell, gene number 2 produce only, yes, good, gene number 2 produce only 1 messenger RNA. If you don't remember it, okay, I am simplifying it. Okay, yes, 2 produce one mRNA messenger RNA. Gene number 2 in this produce, yes, one messenger RNA. Gene number 3 produce one, two, three messenger RNA. Gene number 3 produce only two messenger RNA. This is cancer cell. This is normal cell. So if I say gene 1 in normal cell produce 2 messenger RNA, cancer cell produce 4 messenger RNA. So gene number 1 is overexpressed in cancer cell. Gene number 2 same expression. Gene number 3 normal 3 messenger RNA, cancer 2 messenger RNA underexpressed. I want to compare. What I did, I took normal cell and attached messenger RNA with green fluorescent dye. Which fluorescent dye? Green fluorescent. Green fluorescent dye. And I attach messenger RNA of cancer cell with red. So now I will compare gene expression in two different cell, normal cell and cancer cell. What I did, I took DNA chip. On DNA chip at position 1, complementary probe for these messenger RNA are attached as position 1. At position 1, I attach complementary probe for these RNA for gene number 1. At gene number 1, complementary probe for RNA for gene number 1 will bind. In normal cell, 2 messenger RNA will bind. Cancer cell, 4 messenger RNA will bind. Cancer cell, how many messenger RNA will bind? 4. So at the same site, 2 green, 4 red. So more and more red fluorescence would come. If more and more red fluorescence would come, we will say in cancer cell these messenger RNA are more than normal cell. So in cancer cell gene number 1 is overexpressed because the fluorescence is mainly red color. But at the position of gene number 2, you will find that one messenger RNA of normal, one messenger RNA for normal, and one messenger RNA for cancer cell, because both are equally expressed. So green and red equal amount yellow. So if at a spot more and more yellow color is, mainly yellow color is there, or yellow color is there, means in that mutated cell and normal cell, yes, cancer cell and normal cell express this gene normally. If at a particular site more red, means more expressed in cancer cell. If at particular site more green, more expressed in normal cell. Understood? So this is a global pattern of gene expression. Now you can tell me the answer of this question. Please do reply. To determine global pattern of gene expression in two different types of tumor cell, which of the following technique would be most appropriate for the purpose? Micro